Hello, thank you, Juliana, and welcome and good morning to all of you. Thank you very much for this opportunity to present today. I think that we should get started, and I will share my screen with you. Confirm that you can see my screen, please. Can you hear me? Yes, we can hear you and we do see your screen. Thank you very much. Perfect. Thank you very much. And for reasons of time, I'm going to go straight into my presentation as an introduction, really, to this topic that, as Juliana was saying, is just to set the basis of what genomic epidemiology means, or molecular epidemiology, and what are the key objectives we can use and have with these tools. The first thing I'd like to do, since the title is focused more on genomic epidemiology, but first I'd like to speak in a more broader sense of what molecular epidemiology is, give you maybe some of the background and definitions that I'm sure many of you know, but that we'd like to highlight at this time in our seminar. On the one hand, a very basic definition of epidemiology is the tools that seek to assist in the study of distribution, determinants, and causes of health-related problems in order to prevent and control effectively. And when we speak of molecular epidemiology, what we're actually talking about is integrating the use of techniques and fundamentals of molecular biology. It's important to understand clearly that when we talk about the fundamentals of molecular biology, we're talking about the characterization of nucleic acids as well as the amino acid content, in other words, the proteins, to study the distribution in time and place, as well as some of the determining factors such as transmission, clinical manifestations, and the progression of a disease, as well as how it occurs within a given population. So what we hope to do here is to combine all these tools and understand the distribution of the diseases better. So we have to clearly understand where this comes from. It actually comes from the molecular basis of change. What makes these changes occur to make the actual comparisons? Basically, this comes from the molecular level, from the DNA to the proteins, which is what we'd be somehow calling or understood as molecular. And here, when we speak of the DNA to the proteins, it's important to mention, and here I'm putting this asterisk, the star on the DNA, because we're talking in general of the DNA to the proteins. But when we speak of viruses, for example, mainly the RNA viruses, some go to proteins without necessarily going through the DNA. Some go from the DNA, from the RNA to the DNA, but in general terms, we put it DNA to the proteins, clearly understanding that it could be from the genetic materials to the proteins. You know well, because you've seen this structure of the DNA, and I'm not going to get into the details of this, but I did want to show you the points where something could eventually occur that could generate the molecular epidemiology studies. Basically, in the first process,
I'm sorry to stop you from your meeting.
uh,
If the French interpreters can open the microphone, we're not hearing anything in the French channel. Thank you.
and do I have to do anything? No,
Para
no,
avisarle
no.
al
It seems that the French
Okay.
interpreters had not turned their microphones. Dr. Mendez, yes, please
Ahora
continue.
sí.
So, as I was saying, the one of the first steps when there can be an error or a mutation generating a change is precisely at the level of the DNA replication, which is the first process, which I'm sure you know quite well. And in that process of replication, one when we're copying the cells from the DNA, one can introduce these mutations or changes in one of these nucleotides with an error or a mismatch or a matching that should not happen, and that, if replicated, remains and generates a mutation. The other aspect, the other moment when a mutation or error can occur can be in the RNA transcription. So we have to recall that in the transcription, which is the process through which the DNA generates an RNA chain, which is going to act as a template to act at that level to transcribe the RNA and then translate it to generate a protein. And here in this process, errors can occur in the transcription, which can cause the changes in protein which is being translated. And you know well that there's a genetic code where from these triplets, we're going to recognize the amino acids that are going to form the protein. And of course, depending on the changes that occur at this level, in this template, one can find certain mutations that could change maybe one of the proteins, or in some cases could even, maybe nothing happens, or maybe the amino acids remain the same. In others, it could change from to chemically similar amino acids, or in other cases, there can be even situations in which there are amino acids with different chemical characteristics which can cause even more important changes. In fact, there could even be moments when there are colonies generated where one of the proteins is truncated and is no longer viable. All of this is what forms part of what we can see and should be studying when we speak of molecular epidemiology. We're actually referring to all those changes that can occur at the level of the genome, as well as the impact they can have on the proteins. Thus, as I said, what we're going to try to do in the molecular epidemiology is integrate all these tools and fundamentals, allowing us to characterize the nucleic acids as well as the amino acids and proteins and how they can impact the study of epidemiology. And here, I think it's important for us to mention that though the concept of molecular epidemiology is something that we feel is something relatively new, and that somehow in recent years we began to exploit it more, it is important to understand that this is not really a new concept. This is something that we began to study many years ago. And maybe if we reviewed the literature, we would probably find that the first people who spoke of molecular epidemiology would include people like Edwin Kilbourne, whom already in 1973, in fact, introduced the concept of molecular epidemiology, in this case, to study influenza. It was a very interesting study where they actually began to understand how we can use the different tools from the laboratory and molecular diagnosis with epidemiology. And simply to mention that these first steps of molecular epidemiology were based on the observation of the different pandemics that had occurred, the influenza pandemics. And here we're talking, as if you see on the chart, talking about 1918, which was a great epidemic. And then the later years until the next pandemics up to 1970. One of the observations of this, which is something clearly epidemiologic, of course, and an epidemiologic observation, is that the viruses involved in the interpandemic periods or interepidemic periods only had very small changes at the antigenic level. And when we talk about antigenic, we're actually talking about a change in the protein, in other words, a molecular change. However, and that's something they also comment. Remember that we're talking about 1973. In contrast, they say when pandemics occur, the viruses found in the pandemics of 47, 57, and 68 were actually associated with a radical and dramatic change in the antigenic structure. In other words, we're clearly seeing a molecular finding when we talk about the viral structure, the antigenic part or the protein part, and some impact it's having on the epidemics or the generation of these pandemics. Thus, we begin to review the way in which, or in other words, when we review the pandemic and non-pandemic periods, the introduction of a new virus or a new variant, which at that moment were beginning to be studied. And in fact, it was the first time that they were actually called officially at the WHO. So here we see that in this pandemic, for example, we introduced what was known then as the H1N1. After that, we had the introduction of the H2N2 and then the H3N2. And in these changes, we see that something was happening, and definitely the viruses introduced were different based on the observations done at the laboratory. So what questions did we ask then? And were asked in the study, actually. Why do pandemics vary in severity? And not necessarily lethality, but severity or number of cases. Why do pandemics occur now at shorter intervals? And we must recall now that we're talking about 1973. And where do these pandemic variants come from and how do they arise? And let's recall that we didn't know the viral genome at the time, but we were able to isolate the virus and have it react with cells generating what we now know as the hemagglutination. And this is what was occurring at that point to determine the agglutination capacity or the possibility of actually recognizing the viruses that were arising in the different pandemics. And depending on the antibodies present in each one of the viruses. Thus, we began to try to answer these epidemiologic questions based on the molecular concepts. We have then, and this here is one of the first studies, or I should say observations, that of course are based on laboratories and thinking only of proteins, where we observe that at a molecular level, we were beginning to think of the interaction of the antibodies against the different proteins of the virus, the hemagglutinin and neuraminidase, and how this was actually generating a change or an impact on the hemagglutination capacity of the influenza virus. And added to that, then we were able to observe that depending on the differences in the neutralizing capacity of the virus, there was a definite impact on the severity of the pandemic or the outbreak, which was being seen particularly in the number of cases. And we were therefore able to make an association between a purely molecular finding, which is precisely the change of the proteins and the way in which they're able to be determined, and the impact that they actually had on the viral epidemiology with the studies, which at the time were pretty basic. But at least if we recall in 1973, it was definitely the foundations of what we know today as the molecular epidemiology. And of course, right now, this far, we were beginning to make important studies regarding the nucleic acids. And once we begin to talk about the genomic epidemiology, we begin to refer more definitely to the genetic information, or the information we have from the nucleic acids. And now we were beginning to make the first attempts to sequence and read the DNA as well as RNA molecules. And we were able to establish different types of experiments with different chemical options and different enzymes, where one was able to break the genetic material that was described, and with the different enzymes, allowing us to actually break the genetic material and divide it into those parts that would allow us to see it from different ways. And some studies that were beginning to get done, like Holy in 1965, were able to elucidate the structure of the RNA, a transfer RNA. And a little later in 1972, Walter Fears in fact, for the first time is able to sequence the protein gen of the macrophage MS2, and with different protocols, of course, and different methodologies, some of which included the divisions or gene or bidimensional genes. And then, of course, the most important finding, which was definitely revolutionizing things after many years and after much work in chemistry, was Frederick Sanger, whom I'm sure we all know and recognize, around 1977, when he developed what revolutionized, as I said, the history of sequencing, which were the first sequencing methods using the fragments that were partially digested with the restriction enzymes and were marked with or radio marked with a process at the time called the chain termination method. And for those of you who at sometimes had a chance to work with those methods where we marked with radio marking products, these were the first images we had at the time of the method, which it was known as the Sanger method, and we'll definitely use to understand this. And the genomic epidemiology at that time then began to generate tremendous impact and has actually brought us to the generation of this process, which brings us to what we know today as the genomics. And you have witnessed, I'm sure, all this change and all the progress attained. Here, I just want to mention simple examples where genomic epidemiology allows us to advance in different aspects and objectives we have for epidemiology, which are the causality. In other words, what's the cause of the disease, the pathogenesis, the circulation patterns, the transmission, prevention, or therapy. And in terms of causality, for example, and here I simply want to show you some examples of recent situations which are very clear. For example, this one here of an outbreak of hemorrhagic fever in 2019 in Bolivia, where we only had patients with the clinic and we began to understand the pathogen involved. As background, we had the presence in that area of a virus in 2003, between 2003 and 2004, which was called the Chapare virus. We began to study, and after collecting samples, and thanks to the complete genome sequencing, we were able to determine that the virus that was being detected was different from the one that had been isolated in 2003, which was a new virus. It was a new arenavirus described as the Chapare 2019 virus. In terms of pathogenesis, we simply mentioned, for example, that by comparing the sequencing of genomes, we're able to see or compare, as I said, the genomes that can generate severe or fatal disease. And another example here is methicillin-resistant staphylococcus, which was identified as the cause of a high fatality rate of patients that were suffering MRSA in the study of 2002. And basically through a comparison of different genomes of fatal cases compared to mild cases, we were able to find there were at least 7 genomic islands in the genome, in the complete genome of the bacteria, which were associated to the greater lethality. And that can be seen here very simply, comparing the genomes between the severe and the mild cases. Another with a virus that has been studied considerably, which is dengue. Just to talk a little more about the dengue, the viruses, we see that the Asian genotype of the DENV2 associated to the more severe hemorrhagic cases of dengue. And this is a study that was done in Colombia where it shows that the Native American genotype was replaced by the Asian genotype, the American genotype, which is considered more aggressive. And we observe, thanks to the molecular epidemiology studies, that the introduction of this Asian American strain was introduced precisely at the same time as we saw the first cases of hemorrhagic dengue, as it was known in those times, associated to what was called the pathogenicity. As far as the circulation and transmission patterns, I think we have many examples. And here, because of time, I'm only going to mention them very quickly. We have, for example, the MERS-CoV coronavirus, which you know is one of the viruses associated to the Arabic Peninsula. And here, just to mention, I'm not going to go into the details, of course, but I simply want to show you this very important outbreak of 2015 due to an outbreak in Korea, where as a result of one single case, which is this one here in green, a single case which was in contact with someone, and after several trips in Korea, the person was in different health centers. And as a result of this single case, it not only contaminated family members and patients who were in the health centers, but a large number of health personnel that were in the medical centers that he went to. And this simply to mention that as a result of this, and thanks to the molecular epidemiology, we were able to see that from one single patient, a whole change of transmission with about 185 people was generated, all infected and all in different sectors and different sites, just from one single patient. And that was all determined thanks to molecular epidemiology and the comparison of sequencing. Something similar happened with the H7N9 influenza, where we were able to see that once a virus was described that had never been seen before, we were able to determine in China the genome of the virus. And in a short time, we were able to determine and survey the actual distribution of the virus in China and the way in which it was being distributed. Dengue, as I mentioned already, one of the viruses that has been studied in detail in molecular epidemiology, and it's thanks to these tools that we've been able to determine the origins of dengue in our region. And these are simply some studies that have been done by the CDC in Puerto Rico, where they found the divergence points of the different epidemics of dengue 1 in the region of the Americas. Here we have lineage introduced and which has prevailed in Puerto Rico and in some sites in Central America and the Caribbean, compared to others which can be found mainly in Central America and North America, in Florida to be precise. But here, however, we were able to observe the origin of the viruses and determine from that that despite the fact that these viruses are shared in Mexico as well as in the United States, these are the Nuevo León viruses in Mexico, and these are the ones from Florida. But we were able to determine that though they belong to the same lineage, the origin is different, bearing in mind that viruses in Central America and the same lineage, but from Mexico are completely different, and that the introduction into Florida actually came from Central America with a large number of reintroductions and with different reintroductions into Mexico, where the virus has actually been autochthonous, with only some very small introductions of the virus from Central America, just to show you simply that the arbovirus has also been determined. And this is for in the cases of chikungunya, for example, and how we've been able to determine the distribution of the different lineages and the reintroduction, for example, of chikungunya in the region of the Americas, where the first virus that was introduced in 2013, which led to an explosion of cases in the region, and a reintroduction of another variant, another genotype between 2014, which was found mainly in Brazil. And here to end, simply talk about the genetic characterization of SARS-CoV-2, which is what we've been talking about and working with in the last few months. belonging simply to some work which is the same, but that has shown to date the fact that from the almost 3,890,000 sequences that we've found in the largest database, which is beside, we've been able to determine not only the distribution of a large number of variants, but also the fact that in the evolutionary process that was expected and expectable, we've been able to find a large number of variants circulating and distributed throughout the region. Thanks to these studies, we at this time are able to determine the variants covered in the region and, of course, to see the circulation patterns that we have, not only in the region, but throughout. And this simply to show you a few examples of how we can, as a result of these sequences, get information that allows us to report on the actual distribution of these variants in the Americas. And to end, I simply wanted to mention a few challenges that we still have ahead of us in terms of sequencing on the one hand. Definitely, it is a priority for us to strengthen the genomic surveillance processes. That's exactly the work that we're doing in the genomic surveillance network in PAHO. We have a lot of gaps still. We have a lot of aspects we have to continue to work with. It's still a very expensive methodology, and that's why we have to continue to work to obtain resources significantly enough for the country so that we have the resources so that the sustainability is complex as a result of that. And we also have to think whether it's necessary to have all the sequencing platform in all the countries when often we have different needs, bearing in mind that what we need is actually access to the sequencing capacity where we require different expertise. And that implies also that we need a lot of investment in training and in sampling, which sometimes is not as simple. However, we also have to bear in mind that the sequencing as such is only the first step of what we need to do, and that it's important and necessary to standardize the strategies in order to actually do the genomic surveillance. It's not the same to do sequencing and having sequences than to do genomic surveillance. These are two different things.
Dentro de los sistemas de vigilancia y tener sobre todo objetivos muy claros de la vigilancia. ¿Para qué queremos hacer este tipo de vigilancia? Para, ¿Cómo lo podemos utilizar? Como ya hemos visto, para hacer seguimiento a contactos, para determinar patrones de circulación, para determinar conglomerados en poblaciones específicas, para revisar cambios en la patogénesis y en la clínica, en fin, tener claros cuáles son los objetivos que tenemos para la vigilancia genómica. Y finalmente, simplemente mencionar que sabemos que han habido muchas presiones políticas con las cuales...
I just mentioned that there are political considerations to seek out variants of interest in circulation, but that isn't the main purpose of genomic surveillance, but to look into any variant that may be circulating, which might have an impact or might be involved or can generate information for genomic surveillance overall. So that sums up what I wanted to say by way of introduction to the topics that we have on the slate for today. I thank you all very much. Thank you, Jairo, for that very thorough presentation. I do think that it's very important for us to see these aspects of genomic molecular surveillance. Now, we have a lot of colleagues listening in who are the ones who sent their viruses into the Collaborating Center of WHO, and they have been sequenced, and the genetic characterization tests have been done. And this is important not just for the country, but to understand the composition of vaccines and arbovirus events, dengue, for example. We have two different genotypes of that virus. And so it's important to see how important this is, that goes beyond COV2, which we're all very focused on right now because of the pandemic. And so I'll just open the floor right now for anyone who would like to write a question into the chat, or using a Q&A, or just raise your hand. We will open the mic for you to pose your questions.
Não veo ninguna mano levantada.
I don't see anyone raising their hand.
Ninguna pregunta en el chat.
Nor is there a question in the chat box.
Okay, bueno, pero
Great. Well,
vai estar conectado, entonces,
Jairo will be connected throughout the presentation or this morning session. If you have any questions, please feel at ease to write your question in the chat box or raise your hand. So thank you, Dr. Hector from Uruguay, who says in the chat, everything's quite clear. Now, the next speaker is Natalie Fisher. I'm going to switch to English now.
Natalie is a scientist at Cienzano, the National Public Health Institute of Belgium, and a European Public Health Microbiology Fellow of Cohort 2019 in the ECDC Fellowship Program. She's going to talk to us a little bit today about the genomic surveillance of antimicrobial resistance in Shigella in Belgium from 2013 to 2019. So this is a beautiful example, another way to see how important it is, the molecular epidemiology, to track and survey not only virus disease, but also any other emerging disease. So Dr. Natalie, thank you for participating in the meeting, and we give you the word.
Thank you very much. I will start to share my screen. Okay. I think you can see it. Is that correct?
Yes, we see you and we see your screen and hear you very well.
Perfect. Okay. Well, good morning from Europe and good evening to you. First of all, apologies for speaking in English. I will try to go very slow for the interpreters, but if at some point it's too fast, please don't hesitate to let me know. Thank you for the kind introduction and for inviting me today to present my work on genomic surveillance of antimicrobial resistance in Shigella in Belgium. Oh. Okay. So the objectives of my webinar today will be to give an example of the use of whole genome sequencing for national surveillance of bacterial pathogens. As we have heard already a lot about the use for viruses, that would be a little bit different topic. And I want to focus on the illustration of bioinformatic tools for analysis of whole genome sequencing data. Specifically, I would like to introduce some tools for detection of antimicrobial resistance markers, for the assignment of hierarchical genotypes to compare studies across countries and borders, and also introduce some tools for visualization and annotation of phylogenetic trees. And last but not least, we will discuss briefly some advantages and limitations of whole genome sequencing for surveillance. So let me introduce Shigella to you. Shigella overall are gram-negative bacteria of the Enterobacteria group. There are 4 species known, Shigella flexneri and Shigella sonii, which are the most common. And then we have also Shigella dysenteria and Shigella boydii. And in this map here, you can see the global distribution of Shigella sonii in red and Shigella flexneri in blue. And you can already see quite obviously that the distribution is very different across different countries, where in the Northern Hemisphere, North America, Europe, for example, Shigella sonii is more common. And in the Southern Hemisphere, we have more commonly Shigella flexneri, which is a pathogenic agent mostly of childhood diarrhea. Shigella transmission occurs via the fecal-oral route, which can be through contaminated water or food, and already a small amount of bacteria can cause severe dysentery with diarrhea, fever, sometimes even bloody diarrhea, and stomach cramps. Most cases can resolve without medication on their own within 5 to 7 days, but for more severe cases and also to limit spreading, The most commonly used first-line antibiotics are ciprofloxacin for adults and azithromycin for children. And as you probably already imagined, antimicrobial resistance is increasing and emerging globally and is really a huge problem for Shigella sonnei. For example, in this map, color-coded, we can see that we have Shigella, which express extended spectrum beta-lactamases, or ESBLs, which govern resistance to third-generation cephalosporins, or carbapeneman. We have also Shigella, which are resistant to fluoroquinolones, including ciprofloxacin, which here you can see in the blue color coding. And we have reduced sensitivity to azithromycin, or RSA. And worst of all, you can see here that we have a combination of different resistance mechanisms to different antibiotics, which of course is the most concerning to us. I want to highlight here that over the recent years, antimicrobial resistance in Shigella has been associated in several countries in the community of men who have sex with men, or MSM. Most prominent studies have been published in England, but have also been described in Taiwan, Canada, Switzerland, and Australia. And with this new specific target group emerging, several public health agencies are now starting information campaigns. For example, you can see here a clip from the CDC in the United States about Shigella infection in MSM. Okay, let's switch to Shigella and Shigellosis in Belgium. In Belgium, we have about 400 cases of Shigellosis per year, and 72% are caused by Shigella sonnei. Normally, the clinical laboratories will submit isolates and patient information such as postal code, gender, age, and also severity of the disease to the National Reference Center for Shigella and Salmonella at Cienzano, where I conducted this project. At the NRC, we perform typing of isolated strains to identify the species and the subtypes where possible. We also determine the antibiotic resistance profile, traditionally through broth microdilution in the laboratory. Here, I just want to talk a little bit about the epidemiology of Shigellosis in Belgium across the last couple of years. Here in this epi curve, you can see the number of cases per month, and the color code is coded by the resistance to first-line antibiotics. As you can see in the gray curve, over the last couple of years, we had an increase in cases overall, and we also observed an increase in cases with resistance. Specifically, between 2018 and 19, there was a strong increase of cases with double resistance to both ciprofloxacin and azithromycin. And overall, across the 3 years, 37% of Belgian isolates were resistant to at least one of the first-line antibiotics. If we have a little bit closer look at each of the years and split the cases by gender into female and male, we can see overall that the proportion of cases in men is higher than in females. And moreover, the proportion of antimicrobial resistance among isolates coming from men is much higher. And here I want to point out the spike in double resistance observed between the years 2017 and 2018. And we performed the epidemiological analysis of all cases across the 3 years, and we found that men had an 8.6 time increased risk to carry a strain with resistance as compared to women. So why would we like to incorporate whole genome sequencing into surveillance in Belgium? First of all, currently, it is difficult to differentiate and track strains for public health surveillance or outbreak investigation by the use of classical typing methods, because Shigella sonnei doesn't have any subtypes as compared to Shigella flexneri, and also the genomic diversity is limited. Secondly, as I mentioned, we currently detect antimicrobial resistance through laboratory diagnostic methods. And for example, for the resistance to azithromycin, these methods have only been established in the laboratory in 2017. So we don't have any laboratory data on strains isolated prior. Furthermore, it's difficult for us to know the origins of a strain or identify where it may have been imported from, because information on travel history in the current database is only available for about 10% of the cases in Belgium. So the objective of my project was to describe clusters of multi-drug resistant sonnei in Belgium through the use of whole genome sequencing and to identify associated risk groups. Therefore, we performed first a sample selection. We were not able, due to commonly known restraints, to sequence all the Shigella sonnei isolates. So we selected 372 across the different years and also across the different provinces of Belgium to get the best picture between 2013 and 2019. We furthermore combined our data with data from public databases, 192 samples, coming from different parts of the world, such as Latin America, Asia, Europe, and also with known MSM association. As terms of sample processing, first, the isolates were cultured overnight and then DNA was extracted. In a second step, the library prep was performed using the Nextera DNA sample preparation kit. We sequenced our DNA in 250 base pair paired end reads on an Illumina MySeq machine. And then we used the QIAGEN CLC Genomic Workbench, which is a program which we had available at Cienzano, where we were able to map our reads to a reference genome from Shigella sonnei. We also were able to detect single nucleotide variants and perform a neighbor joining trees, which we constructed. Here, I want to highlight the first method bioinformatics platform that we used, which is called Galaxy. So we have an account here, and it is open source web-based platform where people can upload their data. So you can create an account, you upload your sequencing data, and you have a variety of tools, bioinformatic tools, which are available. And you can see here under workflows, there are already predefined workflows to process your data, but you can also combine different tools into creating your own workflow, which is most suitable to your data analysis. And I think this platform is very nice, and it's meant to be used for people who are not bioinformaticians by training, but who are maybe biologists or epidemiologists, and who would like to use tools easily and fast, and also share workflows with colleagues and get help from the community. So I can really recommend this. And so using this platform, we employed 3 different methods to detect antimicrobial resistance in our whole genome sequencing data, which I'll describe now. So the first tool that we used is called ResFinder, which was published by Bartolaya et al. And this can help to detect the presence of antimicrobial resistance genes. So in our case, we were interested to detect MPHA and Rb, which are able to phosphorylate azithromycin and thereby govern resistance to azithromycin. Secondly, we used the Plasmid Finder tool, which was published by Caratoli et al. And this enables us to detect the plasmids among sequencing data. For example, in Shigella sonnei, we detected a variety of ink-type plasmids, and such plasmids can carry resistance genes. For example, the ESBL genes, or extended spectrum beta-lactamases. And then these can break down and inactivate, for example, cephalosporins. The third tool that we used is called Point Finder, and we used this to detect chromosomal mutations. This tool was published and developed by Tsangari et al. And in our case, we were able to detect various mutations in the GIR-ACE gene in the quinolone resistance determining region. And you can see here that these mutations modify the target site so that ciprofloxacin can't bind anymore, and thereby resistance to ciprofloxacin is achieved. Another tool that we used was used to assign hierarchical genotypes. And this was originally developed to detect and predict antibiotic resistance in Mycobacterium tuberculosis from genome sequencing. And it's called Microbe. This is also open source software available to everybody and is now also available for Mycobacterium tuberculosis, Shigella sonnei, and I think also for streptococci. And so based on this first tool, there was another group led by Kat Holt in the UK, and they extended this tool to be able to perform hierarchical SNP-based genotyping for Shigella sonnei. And I would point you to the paper by Hockey et al., which was just published this year. And so they did this by comparing 4,000 publicly available genomes for Shigella sonnei, and then defining a new nomenclature and a new typing, which enables now to anyone in the world to use this tool and compare their strains and assign the same names and genotypes. So just as a quick example, priorly Shigella was defined into 5 major lineages. See here, lineage 2, for example, Korea and South America, lineage 3, Latin America and Central Asia. And so now, published by different studies, everybody was assigning different names, and it was hard to know if it was really the same strain that you discovered. But now they changed the names, and they came up with a new genotyping framework to easily compare, and I will show you an example in our data set. Okay, the last tool I want to highlight was to visualize and to annotate the phylogenetic tree, and therefore we used R in our studio. So for those among you who are already our users, you may be familiar with the ggplot package. And so the package that we use for phylogenetic trees is called ggtree, which was developed by you et al. And I put here also some links to a really nice handbook and an online vignette for you to have a look. And so with this package, you can upload your tree into R, and it takes many different tree formats, and you can have an Excel table with all your patient data, and then you can match on the sample name, and you can construct your tree and you can annotate it. And I will also show you examples of this in our project. And for those of you who are already our users and who are our learners as well, I can highly recommend the Epidemiologist Handbook, which is a project that I participated in since last year, led by many scientists, epidemiologists, and public health professionals who developed easy tutorials for people who want to learn R and use R in their everyday data analysis. And so as part of this handbook, I developed a chapter on phylogenetic trees, and using the ggtree package, and the data set that I'm talking to you today about was also used to develop this overview. So you're welcome to have a look. It's freely available online. Okay, let's come to the results of the Belgian study. So first of all, I want to show you that I constructed this tree image using the ggtree package. And so here you can see a circular representation, which I personally think is very nice to pick out first impression clusters. So here, each line, as you probably know, represents one sample, and the phylogenetic distance between the samples is shown. In the beige heat map, you can see all the samples that came from the Belgian sample collection, and the ones that don't have a color are the ones from international sample collections. And here you can already see, as color-coded by antibiotic resistance, that we easily identify 3 clusters in dark red, which are both resistant to ciprofloxacin and azithromycin. And furthermore, we have other clusters resistant to azithromycin and also ciprofloxacin only. In the next step, we can overlay more patient data. And so in another heat map circle, we overlay the gender of where the sample was isolated. So in green, you have male, and in purple, you have female. And by doing this, we can already see that 2 of our double resistant clusters are almost exclusively green. So it means they almost exclusively came from male patients. And furthermore, if you look over here, we can see a 3rd cluster which is almost exclusively comprised of male cases, 92%, and only resistant to azithromycin. So this is a very nice method to pick out specific target groups. And as I mentioned to you before, resistance in Shigella sonnei has recently been associated with MSM. So we also tried to obtain this information in Belgium. And you can see here color-coded in red, male cases which identified as MSM as were obtained through interviews of the cases. So for example, here in cluster 1, many of the men identified as MSM. And furthermore, we had 2 samples coming from the UK, which previously were identified as MSM, which were closely related to the Belgian isolates. The same is true for the cluster 2. We had several men identifying as MSM, and in yellow you can see that they clustered closely with cases from MSM clade 2 from the UK. And our 3rd cluster, we didn't have information on MSM from Belgian cases, but 2 different UK clusters, which were very closely related to the Belgian isolates. We can then go even further and we can have a look, for example, only at clusters that are of interest. For example, in this case, we will look at cluster 2 and we will pick it out and we can then represent it also as a linear tree. And here it's easier to see the genetic distance on the x-axis, and you can then also add any information that you want, either as a text. For example, here we added the year of isolation, and you can see that the top cluster with double resistance isolates came from 2018 and 19 and from provinces all over Belgium. And we can then add the heat map, which before was represented as a circle around a tree. We can also add it here in a linear fashion to represent, in this case, gender, MSM association, and various antimicrobial resistance patterns. And if we look at the tree this way, we can see going further down in the phylogeny that our Belgian isolates were closely related to isolates from the early 2000s coming from Vietnam. And moreover, we can see that at this point, if you look at the antimicrobial resistance pattern, there was not much resistance found in these early isolates from the 2000s, but then as they evolved genetically, they also acquired resistance by mutation in the GRAS gene, and they acquired both MPH-A and ERB resistance genes for azithromycin, for example. And now what I want to highlight with the new tool that I described earlier, the microbe tool, we can then assign the genotype. And when we do this, we can see that our previously cluster of interest with the double resistance was of the same genotype as strains that were described in MSM outbreaks in Australia in 2016 and 2019. And more interestingly, we identify also a second cluster, a bit further genetic distance, which was identified as circulating among MSM in the UK. So this new tool is really helpful, even if you don't have the possibility to integrate samples from other countries to compare directly across borders in different studies. And for example, here, we don't know if any of these people traveled to Australia, but we don't need to know that if we have the genotype, as we can see that it's exactly the same genotype as identified in MSM in Australia. So this is really practical. And I don't have time to go into all the different clusters, but I just want to pick out the biggest cluster that we found of 102 cases. And here, this was of the genotype 36112. And this genotype has also been described by Hockley et al. In their recent paper as being responsible for increase of ciprofloxacin and azithromycin resistance in Australia, in England, and in the USA. So we can see that we observed the same pattern in Belgium of this strain, which was circulating between 2015 and 2019. So in conclusions about our Belgian study, I just want to highlight that 1/3 of all Shigella sonnei strains, which were isolated in Belgium between 2017 and 2019, harbored resistance to at least one important first-line antibiotic. Secondly, we found that men have a higher risk to carry strains with antibiotic resistance in Belgium. So this was based on the epidemiological data. And then by including the whole genome sequencing and the molecular epidemiology, we were able to identify several clusters of multi-drug resistant sonnei strains, which were continuously circulating in the MSM community in Belgium, and which were of the same genotype as MSM-related isolates from other parts of the world. And based on our observation, we formulated recommendations for public health, which were also presented to the authorities in Belgium. And we recommended to increase the surveillance, specifically to increase reporting and strain collection across all 3 regions in Belgium, so across the entire country. We recommended to include whole genome sequencing in routine surveillance and to enhance contact tracing of multi-resistant cases of Shigellosis. In terms of case management, we think it is very important to include antimicrobial resistant testing and further improve antimicrobial stewardship. And furthermore, we need to assess the epidemiological situation in MSM in Belgium further through data collection on sexual orientation and through further case control studies. And finally, we recommend to start prevention and education campaigns to make people aware that Shigellosis can also be a sexually transmittable infection, not only among the MSM community, but among the general population as well. Okay, with this, I hope I could show you that whole genome sequencing data for surveillance can be a useful tool to monitor the circulation of strains on a national, but also on an international level, useful to detect a variety of antimicrobial resistance markers independent of the laboratory methods and constraints, and to generate hypotheses about the origins of strains and the history of acquisition of AMR over time. And I want to highlight again, as the previous speaker, that I think open source tools and the sharing of code and data are really needed and are important to bridge the current gaps in surveillance and build global capacity for whole genome sequencing and data analysis. Last but not least, I want to point out some limitations and considerations. So of course, the proper sample collection, transport, and processing is important and is necessary to obtain sufficient genetic material of a high quality to obtain good data. Furthermore, it's important to label and properly identify with unique identifiers your samples so that they can be linked to patient information later. And as I mentioned, in Belgium, the submission of isolates of Shigella is currently voluntary. So if this is the case, you have to be aware that there's maybe some bias introduced due to the non-exhaustive sample collection. Furthermore, missing isolates and data from countries which have currently less capacity for genome sequencing may also introduce bias in our knowledge of the global dissemination of strains and also of antimicrobial resistance. And with this, I would like to thank you for your attention. I would like to thank my collaborators and supervisors at Cienzano and AZG and my fellowship funding from ECDC. And if you have further questions, feel free to welcome me at my email address. I think the slides will be shared afterwards, and I'm happy to receive your questions now. Thank you very much. I will stop sharing.
Natalie, thank you very much for this great presentation. As a virologist, I loved your presentation. It's good for us to see out of the virology world how important is the molecular epidemiology. And I think that indeed, of course, as Sony is an emerging pathogen, and it had a huge mortality, a huge morbidity with the Shigellosis, and how important it is to track this resistance, because if you have a bacteria that is resistant and is disseminating, you have a burden and you have a challenge to control the global spreading of this pathogen. So it was a very enriching presentation, especially when you showed the clusters, how the sequencing is important to see the clusters, the resistance, if it's both, it's only azithromycin or ciprofloxacin, because this will interfere on the dissemination. And I think it also gives basis for public health decisions for how to handle, which measures we're gonna take regarding these pathogens. Indeed, with your first slide, when you showed that male had more infection, more risk, I was going to ask, and then you later showed about the MSM. So how important was the basis for getting to that epidemiological information, even for measures, public health measures? So thank you very much for your presentation. I see that we already have some questions here in the chat. So I'm gonna read it for you. So Dr. Schiparelli, that is the coordinator of the National Influenza Center in Uruguay, he's asking if you could share the link to the Epidemiology Manual Handbook, please. Maybe you can, if you have the link, you can put it in the chat.
I think it was already shared in the chat. I see it already from CDC, Sweden. Yeah, so you should have it there. Thank you.
And Dr. Godoy from Mexico, thank you for the talk. What do you use to do the phylogeny? The whole genome or just a single copy marker gene?
Yeah, sure, thank you. We use the whole genome sequencing in this case.
And then again, that you have a very good presentation. The topic was really relevant. And we have another from US, Dr. Anderson. Very clear, Natalie, very elegant presentation.
Thank you very much. Very kind.
Thank you for sharing with us your experience. I have a little bit of reminders for everyone that are participating. We didn't share, when we want to apologize, that we didn't share yet the presentation and the records of the previous webinar. Our Dropbox, it doesn't allow a large traffic, so we're looking for alternative, but we're going to share. We're going to share with everyone the presentations and the recordings. We also wanted to share with the participants that we would appreciate if you could take 2 minutes to complete a survey to provide feedback to us at PAHO regarding the presentations or any suggestions or comments that you might have, any feedback. And Jessica put it in the chat. We're sharing the chat, the links for the section, the 3rd one on October 5th and the 4th on October 26th. So these links are also in the chat. If you didn't register yet, register, and we'll be very happy to have you again in the following sections. And Martha from Nicaragua is asking if we could share to email with her. So if anyone else have any other question, you can raise your hands also, and we can turn on your mic. I don't see any other questions. Okay, so thank you. Thank you very much, Natalie. And
thank you, it was a pleasure.
And we're gonna have a small break, a 10 minutes break. Now it's 11:05. Maybe we can come back 11:15, and then we continue with the other presentations. So we're back after the break, and now we're gonna have one more presentation about on this focus of genomic surveillance. And who's going to present to us now is Dr. Cristina Ariani. She is the lead of the genomic surveillance operations of COVID-19 at the Wellcome Sanger Institute in UK. As most of you might know, Wellcome Truth Institute is an institute that is a non-profit institute that is focusing on strengthening genomics, not only in UK, but worldwide. And she's going to talk to us a little bit about the large-scale genomic surveillance of SARS-CoV-2 in the UK and the challenge and lessons learned. So, Cristina, thank you very much for being available to participate in our webinar, and I give you the floor.
Muito obrigada, Juliana. I'll turn to English now. Thank you so much for the opportunity to speak at this webinar. I'm thoroughly honored to be here talking about the work that we do at the Sanger Institute on COVID. My intention with this talk is to, at the end of it, have kind of an outline of all the challenges that we have faced along the way. We've gone through a lot and we learned a lot as well. So the idea of this talk is to share with you what we have learned and the challenges that we faced as well that weren't very, there were many actually. Just before I go into my talk, I'd like to present myself. So I'm from Brazil. I have done my undergrad and master's degree at the University of Rio de Janeiro, UERJ. If there are any Brazilians in the audience, you're probably gonna know. Then I moved to the UK to do my PhD at the University of Cambridge, where I've studied Aedes aegypti mosquitoes, genomics. Then I moved to the Sanger Institute to work on genomics of malaria. Pandemics hit, and then we started doing COVID. So I'm going to be talking to you about almost everything, and then please feel free to ask away. The intention is to help as much as possible with the operations of genomic surveillance. So I think I actually have to share my screen. That would be a good idea to start with. So can you see my screen and can you hear me okay?
Yes, we can see you. We can see your screen and we can hear you very well.
Brilliant. Okay. Thank you very much, Juliana. So before I start the actual talk, I just have to read this disclaimer for you, which means that any views are of the Wellcome Sanger Institute, which means in this case my own, and they are not from the UK government agencies, in this case the Department of Health and Social Care or the United Kingdom Health Security Agency. Also, COVID sequencing at the Wellcome Sanger Institute is a component of the national response to SARS-CoV-2 pandemic, and that UK genomic data is generated by multiple providers, not just the Sanger Institute. So talking about the Sanger Institute, what are we? We are a research institute based in Cambridge. We are just an hour away from London, in the outskirts of Cambridge. So it's a very lovely countryside. We do genome research that delivers insights into human and pathogen biology that change science and medicine. And that means that we do large-scale studies on human diseases, including parasites as well. So we do a lot of work on malaria, on cholera, and we have a broad spectrum of genomic surveillance. Hence, we started doing the COVID work. On COVID, so far, what have we done? So just a snapshot of what we've done so far. Over the past 18 months, we have handled 20 million samples at Sanger. That means not just positive samples of COVID, but also negative samples, because sometimes in the past, we used to receive both positive and negative samples. Our current weekly capacity is to do whole genome sequencing of 50,000 samples. So this means that we can receive far more than 50,000 samples, but we can only process and produce usable data for the government for 50,000 samples a week. And so far, we have reported more than 400,000 variants of concern to the UK government. On the bottom of the presentation, you can see that about 70% of the sequencing data, the sequence data generated in the UK was done by the Sanger Institute. The other 30% is from other institutions, which encompasses the COG UK network, COG UK consortium. And Sanger has had about 300 people working on this project. At the moment, we have about 130, so it's an army. So, but believe it or not, these numbers are quite impressive, but it started very simple, just with little post-its on the wall, on the glass wall. And essentially we were just discussing, right, so we have pandemics starting, what can we do? And how do we do it? And what do we do? From the word go, the idea was, how do we produce data that will be extremely useful for public health purposes in a timely manner, which means how can we produce data that is usable very quickly by policymakers and for decision makers. And it was a completely new challenge, not just for us. I think that the whole world was under, well, I don't think it was, the whole world was under the same challenge. And in this case, we were figuring out how to work as a large team to best effort as well. So it was a really quick setup of operations that turned out to be a great achievement for all of us. So this is a picture of our first stand-up meeting that we have at the past we used to have every day. Now we have twice a week, that we still have them. But in the beginning of the pandemics, people couldn't be on campus because we were in a severe lockdown in the UK. And we had to limit the number of people that were on campus. Some people had to self-isolate, and there was, you know what happened. I don't have to explain. But what we did was we asked for everybody at Sanger, for all of the employees, to volunteer. And it was absolutely brilliant because the actual research, the faculty research couldn't happen at that time because of restrictions. But there were very limited number of people that could be on site, and they volunteered to get out of their positions to work on COVID. So it was really nice team effort. So that is the basis of what we call the Project Heron. So Project Heron is the work that we do at the Sanger Institute, the whole genome sequencing of the SARS-CoV-2 at Sanger. So the idea of the project the aim is to do a high throughput genomic monitoring of the SARS-CoV-2 virus in the UK, aiding public health officials in their response to the coronavirus pandemic. We also want to monitor the virus for genetic mutations, which could have public health implications, as I said before. So what is our journey? How we started and where we are at the moment? We started with about 600 samples a week, as you can see here. Then we went to 3,000, 7,000 samples a week, and now we are at 50,000 samples a week. Our failure rate, which means that we have sequenced the samples, but they didn't produce data of good quality enough to be used to inform public health decisions. That failure rate in the beginning was about 30% because we're setting up a completely new protocol, a completely new operations. So fails will happen inevitably. But we worked really hard and we managed to decrease its failure rate 25, 20, and now we are at 15% failure rate. The same as turnaround time. If you remember in the beginning of the talk, I said that one of the crucial points of this project is the timeliness in which we produce the data. So turnaround time, which means is a time in which a PCR test is published. So by the time that the patient knows the results, all the way to the end of the sequencing data being finished, right? So it's this, we call it end-to-end process. So it's from the PCR test to the end of the sequencing results. That used to be 26 days, went out to 19, stayed the 19, and then now we are at 5 days of turnaround time, which is, I'm quite proud of it, to be honest. So sample sequence per week, this is a little bit more detailed information on what the journey is essentially the same plot, the same information, sorry, but so in a different view. So you can see that it's very variable. It's not a steady influx of samples because it varies according to the pandemics at a particular time. So for example, around here in May, there weren't that many positive cases. As you can see, Sanger's capacity was much higher than what we were actually sequencing, and that is simply because there wasn't enough samples to be sequenced. At this moment, there are far more samples to be sequenced than our capacity, but we are at about 50,000. We're not quite there at 50,000, but I now explain why a little bit later. So just a very brief overview of the process. I don't want to spend too much time on this because I want to focus more on the challenges. So essentially, how does it all work? There are 9 labs across the UK represented by these little test tubes and virus in the map here. These national laboratories take the PCR tests. The PCR tests are free in this country. In some instances they are free, and they send the PCR, the whole nucleic acid of each positive sample to Sanger on a daily basis. So every day we receive samples from all the 9 labs, the nine labs. When the samples arrive, along with the samples, there are some data as well related to the samples. we receive them, we process, and what is really important, and I want to discuss a little bit more about it later, is that when we receive the samples at Sanger, there is a checkpoint, there's a decision in which we can make whether we want to sequence that sample or not. And that is because we may want to prioritize some samples. All of us, all our countries, wherever we are in the world, there have been samples of interest floating around. That could be because of a specific outbreak or because of, there is an investigation going on of specific variants, but there are samples that are more interesting to sequence than others. Therefore, at this point, we have the ability to select the samples mostly interested to us. Then we do the processes in the lab that I'm not going to go into detail here, but I'm happy to answer any questions later. And then we have two routes. One is to upload the data to Climb is a data repository where all the sequencing data goes there and is available for the UK government and for the researchers. And in parallel, we report to the UK government, particularly on variants. So on a daily basis of all of the sequences that we have done, we tell the UK government what type of variant that we have found on the data. The excess samples, for example, last week we have received 150,000 samples, and we can only sequence at this point, we are sequencing only 40,000 this week. So there was an excess of 110,000 samples, which then we have 5 temporary refrigerated containers at the car park. Each container can hold up to 2 million samples. The idea is not to hold that many, but it's there for contingency. At the moment, we are destroying samples every 7 days. So whatever has not been processed will be destroyed in 7 days. And we have built some new labs dedicated to COVID work. So here I'm going to talk to you as well about the redundancy and resilience on the process. And because of global shortages, we have decided to have 2 robotic platforms running in parallel. That is because essentially there's a global shortage on tips, and we are struggling to get some of these robotic tips because there's a global shortage of plasticware. And therefore, we have 2 robots running in parallel. If we run out of one type of tips, we have the other one, or we can simply manage the volume that we are processing in each of these platforms. And we currently have 7 Illumina Nova 6, which can process 10,000 samples per machine. But you think that this is about 70,000 samples, which is where we want to be. We want to be at 64,000 samples in a month. So because of the work that we are doing in collaboration with the UK government on the reporting of variants and the timeliness of our response, we have got a lot of attention from the media. And most importantly, we have produced very important data for public health purposes. For example, here on the Delta variant, the use case of the Delta variant, at the end of March, here on this plot, you can see that in yellow, it means very low prevalence of the Delta variant. And then 1 month later, we started to see a little bit of a cluster. Another month, 2 months later, the incidence, as you can see here from the heat map, the incidence has increased considerably. And 3 months later, the country was dominated by the Delta variant. And we could see this spread, and we could see this very clearly because of the whole genome sequencing. And we were able to detect this very rapidly because of the good coverage, geographical coverage. And this data is available in this website that you can see there on the bottom. There's far more data than I've presented just now, so you can select the different variants. you can see the incidence, and if you go there and if you press play, there will be a little animation of how it varied on the map. And it's really interesting piece of data. Right, so this is the part that I wanted to take some time and discuss with you, which are the challenges. Because any lab, anybody that will be doing some whole genome sequencing of any, for genomic surveillance purposes, will face some of these challenges. One of them, for example, as I said before, is the global shortage of supplies. Not only plasticware, but all of the reagents, not all of the reagents, but a proportion of the reagents that we use to produce whole genome sequencing, there is a global shortage. Therefore, we have to manage expectations, and we have to manage the production as well. So we do have at Sanger the capacity to produce to sequence 50,000 samples a week, but we are not there. We're not doing it. We're doing 40,000 because we need to manage the reagents and the consumables we have. Another challenge is the constantly changing landscape. And this is, in the pandemics, is the norm for all of us in our personal lives, I suppose, and also at work. This project has changed landscape almost in a daily basis, and that is because of the nature of the virus is always changing, and there are always new things, there are always new pressures, and we need to respond to the government very quickly, a new report, a new technique, and many new things happening all of the time. And it is challenging, it is challenging. Another challenge is the logistics, because one has to get the samples to the institute. And at Sanger's example, the logistics challenges is that we have 9 different labs sending samples to Sanger. Bear in mind that the UK is far smaller than Brazil. So if in Brazil, this and other countries as well, I'm thinking of Brazil because I'm Brazilian, but this challenge is even greater, I suppose. And not just the samples, but also the data that accompanies the samples, because there's no use if you receive only the sample, you need to have the metadata associated. You need to know on a plate which one is the sample in each, so in each well. So you need a plate map. You need to know what is the boxes that you're receiving, what is in each of these boxes. And also you need to know some other metadata, for example, the postcode or part of the postcode of the person, so you can geographically identify. So there are lots of logistics that needs to be dealt with, and it is challenging. Another challenge is the sheer volume of samples handled. There was 1 month in July that we received 181,000 samples. And it was very challenging. And even other labs that don't receive that many samples, there will always be questions to receive more. So, regardless of the sheer number, the absolute number, but you're always going to be asked for more, to do more, to receive more. And that is a challenge because you need to manage expectations. This is a very large teamwork, and that comes with itself with challenges as well, because you have to manage people's expectations, you have to manage the whole line. In my case, my group with Sonia Gonçalves and Alan Keith, who might not be here at the moment, I don't know. We look at the end-to-end, so we are responsible for everything from sample reception to the reporting to the UK government. So it's a lot to work on, and we work with several teams. And even if you don't have to work with several teams, there will be a large team involved because of the amount of work that genomic surveillance needs. There will be external dependencies. As I said, there is data needed from elsewhere. And in our case, we automate our processes, and an automation is very strict. The more automated things are, the less jeitinho, as we say in Portuguese, which means less room for less room for adaptation there would be. So this is an issue. Another one is the timeliness of the outcome. Ideally, genomic surveillance has as near real time as possible response. Therefore, the closest to reality, the data and the results, the better for public health purposes. As I said, managing expectations. So everyone wants their samples sequenced. The researchers, the governments, because everyone will have a valid reason to want their samples to be sequenced. And sometimes it is very hard because you have to say no to some people. And sometimes it's just impossible because we have too many and there are too many logistics issues. So for example, we have developed, because of this, we have developed a sample prioritization algorithm, which allows us to deal with outbreaks, with sample of interest, as we said. And that allows us, I have a map here to show you, that allows us to have a good spread of the samples in the country. So what this algorithm is doing is that it's prioritizing some specific samples. At this point in time, we are very interested in imported cases. But also it's ensuring a geographical coverage of the UK, because this is very relevant for contact tracing, for example, and for understanding the pandemics. So this sample prioritization is, I really like what Jairo said in the beginning, which is the interesting part of this project as well, is that these type of data allow contact tracing. And in fact, in February, where the P1 variant was starting to become prevalent here in the UK, I was manually producing reports and giving data to the UK government on a daily basis, because that data would trigger a whole load of contact tracing. And they would send, if they found a P1 variant, for example, somewhere, let's say in Cambridge, they didn't, but in Cambridge, they would send mobile testing units to the area, and they would do a PCR test in the neighborhood. And then the positive samples would be sent for sequencing. So, and that sample prioritization allows some of these, outbreaks and special samples to be prioritized at sequencing stage. So we had another challenge, which was internal reporting. We need to know where the samples are in order to improve the process, right? So it's not just communicating things to the government, but also it's always looking inwards into our own process. What can be improved? What can we do better and faster? And so we had to build a new system to track samples internally at Sanger. And as I said, there was also the challenge of external reporting to the Department of Health and Social Care. We didn't have anything in hand, and we had to build these reports. In February and March, I spent 2 months from Monday to Sunday, every single day, producing reports manually. And then now they're fully automated, and they go automated directly to the government. And so it was very challenging to build everything from scratch, but very rewarding at the same time. Another challenge that I think is not just to genome surveillance, but is for the whole world, which is staff self-isolating. Obviously, during the pandemics, this is going to happen, and then we might have a limited number of staff available. And believe it or not, we had to face flood, we had to face 2 fires, we faced many, many hurdles in the buildings as well. There was a lot of sweat and tears, but also some laughter. And to be fair, a lot of laughter, because you have to. And this is my last slide. What have we learned? What have we learned out of all of this? So I think one of the most important thing on this is to develop a really good relationship across all parts of the end-to-end process including external organization. We have a really good relationship with the Department of Health and Social Care. We have really good relationship with the Scottish government as well, with Welsh, and this makes, you know, everything more efficient and robust as well. Another thing that we have learned is not to focus on target. So we never said that we needed to be at 5 days, but we've always, the aim is to be as close, as short in turnaround time as we can, you know, in a realistic way. So what can we do to improve? This is our constant question. What can we do? Is there any tiny little bit that we can improve? And that, you know, this is really important. Another thing is that work really hard at creating a single view of the process through performance data. So it's end to end. So without too many arms and branches. So simplify the process. And as you can probably have gathered from the challenges I've mentioned, expect bumps in the road, bend and flow with them, and seek improvement over a long period of time. The improvements don't come from 1 week to the next, but they will come over time. Continually invest in the science and in robotics and support functions. So there's a lot of unsung heroes in this talk. So there is a huge amount of IT that goes into this, and also facilities and human resources. And there's the full amount of, you know, the full suite of people that support the project. Another lesson is to recruit lab leaders that are used to high throughput labs, or if you're not doing a high throughput lab, they are used to the setting that you want to set up. Always develop more capacity than you need. So then you can cope with issues and problems. For example, this week we have one reagent that is not, we don't have too much of it, but we may have you know, a different type of reagent that can substitute. So we have to create resilience, as I said in the beginning, with the robotics as well. The data QC part, which looks at the end sequencing data, is crucial and is really important, a really important part of the process, because this is where the data is going to go, and high quality data is paramount. And to finish up, difficulties were in the constantly changing landscape, as I'm sure you have gathered from everything that I've said, and external dependencies. We depend a lot on what other people supply to us, other organizations. As it was hard to pick apart what changes were having impact, and as often another part of the process would also be influxed. What this means is that we depend a lot on the data and on the samples given to us, and that is challenging and it's difficult. So the more you trust the data and the samples that you receive, the better. So what else? Excuse me. What else do we have to do? We just excuse me. We just have to keep improving, increase capacity if possible. And who knows what else is going to come. I'm sure more will come as well. So I'd like to say a huge thanks to the Department of Health and Social Care, the genomics program within the department, the project Heron, the COVID team at Dengue, which is huge. Unfortunately, I cannot name everybody, but particularly Dominic Pratkovsky, John Silito, Sonia Gonçalves, as well as the Parasites and Microbes Department at the Dengue Institute. So we do have a Wellcome-funded PAHO-led initiative to establish genomic surveillance of COVID in Latin America, and that involves Josefina, Enrique, and Nicholas Thompson on that work. And we have been funded by the Wellcome, DHSC, and NHS Test and Trace. And with that, thank you very much.
Muito obrigada, Cristina. Thank you very much for your presentation, for sharing all the challenges and the lessons learned, because indeed it is an unprecedented pandemic that we're going through, and there have been a lot of challenging inside Ministry of Health, with EPI colleagues, inside public health institutes, inside the laboratory as well. So it's, I think the way you presented and you shared your experience, we felt familiarized with it because it also happened to us. And I think it's not only the staff self-isolation, but it's also the lack of human resource, because we had a surge in the demand of processing samples, of giving results, of responding. And this reflects on public health and on surveillance, because surveillance has to be timely. If I'm processing a sample that's from 2 or 3 months ago, I'm telling the story of what happened during the pandemic. I'm not doing surveillance anymore. So this is a real challenge when we work all together in this big puzzle that is public health response and emergency response. And I really enjoyed to see how you could drop your time from 26 days in April to 5 days in September for giving results. So indeed, I have a question. I see that some colleagues also have questions in the chat, but this would be inside the laboratory. Of course, here in the region for us, it takes more time, about 14 days since the samples arrived at the laboratory. But I wanted to ask you about the shipping of the samples. Of course, you're in the same country, it's easier. How is this logistic? And also just to add about the criteria as well, because it's not only to have a case, I want to track a case that has been a contact or not, but the quality of the sample is also important for the sequencing, including the viral load inside this sample. So if you could just let us know a little bit about this, this shipping of the samples to the Institute.
Yes, thank you very much, Juliana. Yes, so the samples are shipped on a daily basis, and it used to take about 5 to 6 days for the samples to arrive at Dengue. So we hired a few people to work in these labs and to help us understand where were the critical points that could be minimized. So there was a lot of work done at the lighthouses and by the lighthouse labs, which is where the PCR tests are done, to reduce this time. Today is about 1.5 days between the test taken. It's not the test taken by the person, but it's by the test result. So it's when the PCR test is done to the Sanger Institute. So essentially, they do the PCR, and the material left, which is whole nucleic acid, they cherry pick. So they transfer only the positives into a plate, into an 96 deep well plate. They will put these into boxes, and they will ship in a -20 refrigerated van to the Sanger. And this was one critical point for us to decrease the time. In terms of sample quality, that is both are really good questions. Sample quality, we don't sequence anything that has CT value above 30. So the PCR test, the qPCR test, produce the CT value, and we have seen that if CT values are above 30, the chances of not having very good sequencing data is greater. So we decided not to sequence those. It's not worth it, unless there have been cases, particularly at that P1 time, that there were some samples with very high CT values that we sequenced regardless, and we didn't get good results. So it is very important to have that sequencing quality a priori. But it's not so easy because this is the maximum that we have. It's the volume, so the amount of sampling in the plate, as well as the CT value. But we don't have much more than that to assess sample quality prior to sequencing.
Thank you. And we have some comments and some questions here in the chat. I'm gonna read it for you.
Thank you.
Here. The first one is from Dr. Gotti from Mexico, Godoy. How many samples are sequenced per run? And what sequencing deep it indicated to the variant analyses? It's a titanic job. Thanks for sharing.
Thank you. Yes, so in a NovaSeq run, there are 2 lanes, and each lane we put 376 samples. Essentially is a 96 well plate put together, but there is 2 controls per plate, so 8 controls in total. So in one run, there will be 2 times 376 samples. And the sequencing depth to do the variant analysis is 50 times 50x coverage.
And then there's another question from Dr. Ingrosso from US. Great presentation, very impressive your effort. And again, more compliment, Dr. Salazar from Guatemala. Great presentation, muito obrigada. Dr. Serpas, to upload your sequence data and make them public, what were the main issues you had to run the QC analysis?
The main issue was, again, the timeliness. So before we were doing manual QC, and then we used the pipelines that we had for our previous work at Sanger that was already well established. But then we developed an additional pipeline that is auto QC. So essentially the sequencing run finishes, it triggers the QC pipeline automatically. If that pipeline looks at the data and thinks that everything is kind of normal, and by normal I mean no samples are, show any weirdness, or for example, most of the samples have equal representation. There is no gaps in the genomes that triggers some red flags. So these red flags would be automatically triggered, and if they do, they will go into the manual QC. And the difficulties is just making sure what is a good quality, because you can be as stringent as you want, and it's figuring out that level of stringency that for you and for the UK government and for the community as a whole, it means that it's good enough. And in the beginning, we had to figure out with the community, that was the COG UK consortium, that there was a lot of discussions on what it was acceptable or not, obviously. And then we build our pipelines. In a high throughput setting, the QC is very important in terms of cross-contamination. The larger your operations, the more careful one has to be regarding cross-contamination of samples, simply because of the amount of things that are going in the lab. So this was something that we were always looking at very carefully as well.
And then, thank you, Cristina. And then there's Dr. Bonsi, that's the director of the Public Health Laboratory in Haiti. Thank you for the great presentation based on experience. Also, Dr. Lerman from Argentina, also thank you for your presentation. A reminder for everyone to please don't forget to register for the next 2 webinars. So the link is here in the chat. More accomplishment for the presentation. Dr. Gelé, also from the National Public Health Laboratory in Haiti. What about the maintenance of the instruments? If you can share a little bit of your experience.
Yeah, that is a really good question as well. So we had some issues like a month ago that 2 robots started failing, and then essentially it was overused. So we had to review the maintenance of the equipment, because it was together with the ramp up. So as we ramped up, we had to increase the maintenance of the instruments. And that is another reason why it's good to have redundancy in the system. So as we ramped up, as we increased the number of samples that we receive and produce and process, we also started producing redundancy in the system, to allow for instrument being maintained or to allow the instrument to break down. So is for contingency as well, but that's a very good question. Instrument maintenance is something that we have to be very careful about, to making sure that the frequency is optimal and there is minimum impact as well.
And then there's a question here from Dr. Cepas. Do you use any Rgbtr protocol as a variant screening test before you run the sequences?
Yeah, that's a very good question. So we don't. So what happened in the UK is that some samples go, they fork. So they go into two separate ways. So the same sample can go to whole genome sequencing, or then can go to something called reflex assay, which is an RT-PCR type. So the reflex assay looks at only specific mutations. So, but we can't wait for this, because that would introduce about 3 days in the pipeline, or even probably even more, about 5 days in the pipeline. And there's all the logistics to say, oh, this sample is this variant or that variant. So at the moment, we don't do, at Sanger, we don't do any RT-PCR prior to sequencing. But there is this line, this deviation of some samples go through both in the UK.
And
Thank
then
you.
here there's one more question from Dr. Cruz from El Salvador. Good morning. For measuring the quality of the RNA in the sample, do you only use the CT value, or do you use spectrophotometry or any other methodology?
Yeah, we only use CT value, and because we don't have we can't do anything else at this point, because yeah, it's the data that we have available, and it's the only thing that we can measure.
Perfect. And then the last accomplishment of an excellent presentation. We could really see ourselves reflected in your presentation, of course, with some differences that we have between the regions, but it was a really good presentation. So Cristina, thank you very much for your time and for sharing your experience. If anyone has any other question, want to raise your hand or comment. I don't see any other hand. Okay, perfect. So thank you very much, Cristina, for your presentation, for your time to join with us. And then I think we're getting to the end of this webinar that was very enriching. And I'm gonna give the word now to Andrea, that is the advisor for epidemic-prone disease, epidemiology. And so we started with laboratory, and now Dr. Andrea, that is also the team lead, the influenza coordinator for closing the webinar. Andrea, you have the floor.
Thank you so much, Juliana. Thank you so much as well for coordinating today's webinar. I will switch to Spanish if possible. Nada más para agradecer seguramente a todos los participantes.
Thank all participants for taking their time to be with us here in this second webinar. What we've thought was to maybe extend it to a fourth, so that we can have a more limited duration. And that's why I'm not going to take up too much time in my words to you, to remind you just that we're going to have a third seminar next week on October 5th, and another one we've added, as I said, on the 26th of October, so that we can complete the sessions in 2 hours like today. The links to register are in the chat. Take your time, please, to register. The truth is that we want to have these seminars quite open, but we also have a target audience that we'd like to work with to develop this topic, not only for the webinar, but for long term. And We're considering 3 profiles that we'd like to have participating in the next webinars. 1, technical professionals from the National Influenza Centers or the reference laboratories for influence COVID-19. 2, as we've said today, is the epidemiologists at a national level who are responsible for surveillance in general and the respiratory codes. And the third group is the professionals from the Health Ministry who are responsible for the preparation of early detection and response. Actually, the idea of this webinar is really to stress what are the sequencing, molecular sequencing capacities which are expanded normally, and not only within the laboratory environment, but in what was the epidemiology of national surveillance health systems. And the third item is we're trying to resolve the situation in a way that you can access the presentations as well as the registry of these webinars. And I hope that we can keep in touch as well on this. And finally, thank you for the contributions, Dr. Cristina Ariani, Natalie Fisher, and our very own Jairo Mendez in your presentations today. I want to thank those who helped us in organizing the agenda, Justina Valencia, I'm sure, and her team, Juliana, Leonel, Patrick, Jairo. And lastly, but not least, I'd also like to thank the colleagues who make it possible for us to hold these events in the logistics. Jessica, Judy, Marta, Lee, Manuela, Carls. We want to thank you all for your support, for making this possible. And of course, the interpreters, Ronnie, Brian, Susana, Claudio, Mirna, Silvia, Pascal, and Amelie. Thank you all very much for participating and for your contributions. And we'll see you on October 5th, same time, and we'll be sharing the agenda a few days before. Thank you very much. Have a good day, and we'll see you the next one.